Hello everyone, good morning. Today is Tuesday, March 9, 2021 and it is 9 a.m. Eastern Standard Time and this is the start of our program for virtual online ART training. Today we are going to cover Module 2, Advanced Andrology Technique. My name is Ashok Agarwal. I am the director of the Andrology Center. I welcome you all to this program. We are very, very pleased to have you join us. And let me begin by my introduction. Okay, so I'm going to be your host for the next 2.5 hours. So let us begin. This is a brief biography about myself, and without taking too much time, I'm in charge of the Andrology Center here at the Cleveland Clinic and have been there for many years. I'm also the director of the American Center for Reproductive Medicine. I staff in the Department of Urology. We are very proud as a group. Our program is recognized as #1 in terms of the number of publications, the quality of publication, and the reach of our publications in the field of male infertility, andrology, and human-assisted reproduction. We have a very large number of medical textbooks that have been published. And we are very proud that very large number of clinicians, researchers, students have received training in our program from more than 100 countries over the last several years. I want to declare that the organizers and speakers of this program have no conflict of interest. We have not contacted any companies or outside entities for sponsorship. The program that we offer is entirely made possible by support from our center. Webcasting is supported by David Reichling and David Wolfley from the Information Technology Division of the Cleveland Clinic. Thank you to both of them. Further, the course faculty have donated their time for this online training. The program coordinators have volunteered their time for this training course. The names, images, and description of certain products or instruments shown or discussed during our presentations are being used routinely in our Andrology Center. These items are not endorsed by the speakers. Now, let's talk about what we are going to be doing today. So today we are going to be teaching and training you in advanced andrology techniques. And the techniques that we are going to cover, we are going to start by discussing what is advanced sperm function test. Then we are going to go to the most important test which is currently being employed, is called sperm DNA fragmentation test. We'll also cover test for oxidative stress as oxidation reduction potential test. Then we are going to talk about the lab developed test for sperm DNA fragmentation, and then cover the clinical relevance of sperm DNA fragmentation in human assisted reproduction. And lastly, there will be a talk by our clinician on the clinical interpretation of everything that we are going to be covering here. So these are again the topics from starting in a clockwise manner: a laboratory evaluation, clinical relevance, evaluation of SDF, and lastly clinical interpretation of laboratory tests, laboratory results that you will receive, and how to make sense of it. And this is the program in a on the left side, and our speakers for the day here. Our speakers are Professor Ralph Henkel, Dr. Sejal Gupta, Dr. Renata, Dr. Rakesh Sharma, and Dr. Neil Parekh. So today we are going to be talking about the advanced andrology techniques. On the left side of the panel, you see the basic test, which can be covered under the semen analysis and many of the ancillary tests that you heard last in the month of January during module 1. So these are basic tests. And then we are talking about the advanced andrology test. And there are a variety of tests. This panel has just a small number of tests, which we have illustrated here. And many of them, or some of them, will be highlighted in today's talk. So why we chose the test for oxidative stress? And the reason is that there is such a large amount of information on the role of oxidative stress in male reproduction and male fertility. Right from the very beginning, from 1940 to current, there are thousands, almost close to 1,200 publications, which is probably the largest number of publications in one subject matter. So it is a very important area, and we felt that it is important for us to cover that. And also we, on the right side, you can see we have a great deal of expertise in this particular area, and we feel that we can do a very good job in talking about and explaining the practical side of these tests for male infertility. And then the sperm DNA fragmentation, another important test. So if you look at the left side, again, this is a very hot area with very large number of publications, both original publication reviews and meta-analysis. And this data is only until 2019, and certainly the number have increased since then. And on the right side, again, we have at the Cleveland Clinic a great deal of expertise in this area, not only from a research point of view, but also from a clinical point of view. We have developed techniques, we have standardized those techniques for sperm DNA fragmentation testing, and we felt that we can share these things with a great deal of expertise and confidence. Of interesting findings and results, not only from our group, but from many other groups, which she was able to explain. Our next speaker is again Dr. Sharma, and Dr. Sharma is now going to speak on evaluation of sperm DNA fragmentation, or also called as SDF, by lab developed test, or LDT. So he's going to take it over from here. Rakesh, you can begin.
Thank you, Dr. Agarwal. Good morning. This is going to be my second talk. The learning objectives are the TUNEL assay by using a flow cytometer, understand the principle of flow cytometer, and more important, I'm going to highlight the terminology used in the measurement of sperm DNA fragmentation by flow cytometry, and also highlight some of the lab scenarios as well as the quality control in using sperm DNA fragmentation by flow cytometry. In the last 20 years, the 4 commonly published tests are illustrated in this figure. And as you can see, sperm DNA fragmentation testing by TUNEL is the most widely published technique in the world. And sperm DNA fragmentation can be measured by 2 methods, using the fluorescence microscopy, in which we measure 200 to 500 sperm. This method is subjective, time-consuming, and many times inaccurate. The other method that is used is the flow cytometry. And in this method, 10,000 sperm are scored. It is objective, rapid, robust, and a very sensitive technique. And therefore, today I'm going to talk about the measurement of sperm DNA fragmentation using the flow cytometry method. The principle of flow cytometry is that the TUNEL method measures both the single as well as the double strand DNA breaks. It's a direct method which tags with the enzyme binding to the sites which are broken or which have the 3-hydroxyl damage, and these can be measured using the flow cytometry. On the right is the figure giving a short protocol which has been discussed and shared with all of you. Here I would like to spend some time to highlight the principle of flow cytometry that we use in this benchtop flow cytometer, which is called the Accuray C6, and this is available through Becton and Dickinson. The basic principle of the flow cytometer is that the sperm are passing through a sheath fluid into single cells, and these are then excited using a blue argon laser, and the light that is reflected allows us to measure two important components, which is the forward scatter, which measures the size of the cell, the side scatter, which measures the granularity of the cell, and then based on the stain used for the tunnel, the absorption is by the green filter, which is the FL1 detector, and the propydium iodide, which is measured in the second filter, which is the FL2 filter. And here it shows the FL1, which is used to measure the green dye, which is FITC in this case, and the FL2 measures the red absorption, which is coming out from the propydium iodide used for staining the nucleus. The signal is then amplified and then converted into a light signal, which then can be visualized on the computer. The difference between the benchtop C6 flow cytometer and the conventional flow cytometers is that this flow cytometer uses a peristaltic pump instead of the voltage detectors which are seen in conventional flow cytometers. This figure illustrates the kits, the reagents that are used, as well as the positive and the negative controls that come with the kit used for measuring the DNA fragmentation. Here I would like to spend some time in familiarizing you with some of the important terminology that is used when measuring the sperm DNA fragmentation using the flow cytometer. So this space that you see on the computer when measuring the DNA fragmentation is called the workspace. This allows us to measure each sample, and we provide the information over here. This is the plot, and on the screen you can create your own plots, 1, 2, 3, 4, 5 plots. And then we have the gating, which is the area that we allow for the cells to be measured. Anything outside this gate will not be recorded and measured by the instrument. This is also called a template. We create a template both for the kit as well as the test samples, and all cells will be measured within the template. The workspace is this area, and this area will allow us to not only record the information about the sample, but it also has the settings that we use. And the settings over here are that we run the sample measuring 10,000 events. We measure the P3 in P1, and what this means is that we are only capturing the cells which have complete nucleus, and we're excluding the cells which have apoptotic bodies that give us erroneous results. The fluidic speed is set to slow speed, and the threshold is set as 80,000. We also have the last plots showing the quadrant, and these are the 4 quadrants, the lower quadrants and the upper quadrants, and the extent of DNA damage is indicated by the upper right quadrant. In the flow cytometer, which is BDC6 Accuray, we make use of the virtual gate alignment, and in this case, a standard sample with a DNA fragmentation of less than 17% is used as the reference sample or the standard sample, and all test samples are then aligned with the standard sample to give us the final reading. And this is called the alignment strategy, and it is very important when running the samples for DNA fragmentation. So Having understood these terminologies, I will take you to the next step. And this slide shows the most important step, which is the sperm preparation. For measuring tunnel using this method, we require a minimum of 2.5 million sperm. We also include a negative control, which is the samples in which we do not add the TDT, so there are no cells which are stained for DNA damage. We also include a positive sperm sample, and in this case, the positive sample is created by using hydrogen peroxide and incubating the sample for 1 hour at 50 degrees Celsius. The samples are permeabilized in 3.7% of paraformaldehyde and fixed before they are analyzed. Quality control is the first and the most important step before we even run the test samples. And this is important because the test measurement is an elaborate process, and it is important to know that the setting of the instrument as well as the reagents and the staining is optimized, and the quality control for the C6 is accomplished using the 8 peak validation beads. And essentially these beads provide us the 8 different peaks which can be measured for the 3 detectors, detector FL1, FL2, FL3. And the first plot shows the area where the events are tracked. We measure only the singlets and we exclude the doublets or the cells which are sticking together. The second plot will show the 8 peaks that are generated for each of this detector FL1, FL2, and FL3. And we measure the peak of the brightest fluorescence intensity, which is shown over here, and also check the coefficient of variation. The coefficient of variation should be less than 5% for the quality control to pass this criteria. Here I would like to show a quick video for the quality control. We take 500 cc of water and we add 4 drops of the quality control bead. Here is the gate, and we highlight the well in which the measurement is taken. We place the tube with the beads and we set the gate setting to 50,000 events. And here we are measuring the cells or the events, and these should be more than 70%. And here you can see the beads that are showing the 8 peaks, and we measure the fluorescence intensity of the brightest peak, and in this case it is 410,000, and this is showing the coefficient of variation of 1.49, which indicates that this quality control has passed the test, and now it is good to test the samples for DNA fragmentation. We make use of the kit control, and kit control contains the cells which are different than the sperm cells, and therefore the gating strategy will be different for the kit control compared to the sperm samples. And this plot shows the measurement of the negative and the positive kit control, and the quadrant for the negative kit control will give the value of 1.1%, which is negative for sperm DNA fragmentation because this is a negative control. Similarly, we will see the damage of 45.2% in the positive kit control, indicating that the 2 cells or the 2 samples are providing very distinct DNA fragmentation, and now we can measure the DNA for the test samples. For the test samples, again, the template creation of the template is very important, and once this template has been established, we always use the same template every time that the sample is run. The settings are set to 10,000 events. In P3 in P1, which as I explained earlier, we measure only those cells which are stained for the nucleus. The fluidic speed is set at slow, and we set the threshold to 80,000 events. and the measurement is done in the collect tab. It's very important to make sure that we are performing the test in the correct settings. So the measurement or the samples are run in the collect tab, and this workspace illustrates each well representing a single sample that is being tested. And these are the plots where the events will be recorded. We create this gate to exclude the debris from here. And the second gate indicates that all the cells in this gate are the ones which are having proper sperm nucleus, which are stained and are being measured. And the quadrant will actually give us the extent of DNA damage. In the collect tab, we also make use of the standard semen sample, as I explained, which has negative sperm DNA fragmentation, and each sample is then aligned using the virtual gate setting. And once this is done, all samples will be aligned to the standard sample, and the measurement will be done in the Analyze tab, where the 3 detectors are represented and will provide us the final value of the sperm DNA fragmentation for that given sample. And to illustrate this, I will show you a quick video clip. So here we see that the workspace is the unanalyzed. So here we have run the samples, but we have not analyzed the samples. And we are checking the distribution of the events in the gates. And here we make use of the standard sample, as I mentioned, and we will use the virtual gate setting to make sure that all test samples are aligned with the standard sample. And this is how the alignment is done. This is the standard sample. We place the marker right in the center of this peak, and then we use this to measure the alignment for the test sample. We pick out one test sample and we align the blue line over here to match that with the standard sample. So this is called the alignment using the virtual gate setting. And once the alignment is done, we will see an asterisk or a star, which indicates that the test sample has been aligned with the standard sample. This is very important because if the alignment is not done, then the distribution of sperm DNA fragmentation will be incorrect. Once this is done, we go to the Analyze tab, and in the Analyze tab, we plot the 3 detectors, and these 3 detectors are then shown over here, and we check the test that we are measuring, and this will provide us the events in plot 1, plot 2, and plot 3. And the plot 3 is the one where the sample has been analyzed, and the upper right-hand quadrant will indicate the extent of DNA fragmentation, which in this case is 18.6%. Each sample will be shown in the Analyze tab, and the last quadrant will indicate the extent of DNA damage. For example, the second sample has a DNA damage of 0.6%. So this is how the sample is analyzed and final results are documented. There are many factors which can affect the results. Number 1 is the accessibility of the stain to the DNA. Number 2 is the sperm preparation. Any errors or incorrect preparation will lead to erroneous results. The presence of dead cells can also impact the DNA fragmentation, the number of cells examined, as well as the inter and the intra-observer variation, and the inter and the intra-assay variation must be established within each lab. The figure on the right shows the receiver operating characteristic curves, which allow us to establish the cutoff. In this case, it is 16.8%, as well as the sensitivity and specificity of the test. And the figure on the right shows the distribution of the DNA fragmentation in the controls as well as the patients. And as you can see from here, all controls have a value less than 16.8%, whereas the patients show DNA fragmentation ranging from 16.8% to as high as 40 to 60%. This is how the TUNEL report is generated. We provide the information for the patient, the lab where the test was conducted, patient's information, and the test that was used, in this case, the flow cytometry using the C6 analyzer, and also the reference values for the normal as well as abnormal sperm DNA fragmentation, the display of the positive sample or the actual values of the sperm DNA fragmentation, and the results are interpreted for the clinician. Instrument quality control is extremely important in the measurement of sperm DNA fragmentation, and the quality control can be the different fluidics that are used, and these are the reagents used for the decontamination, cleaning, extended flow, as well as the bacteriostatic solution. As I mentioned, it's a peristaltic pump, so we have to be very careful that the fluidics is correct, the capillaries are not clogged, and these filters are changed at a regular frequency. Many times, the clogging can occur in which it will slow down the measurement of the events. And to troubleshoot, we place these fluidics in warm water, and then we flush the cycle, and this will clear the SIP and the measurement can be done. When we perform the sperm DNA fragmentation using the flow cytometry, there are many situations which can occur, and each of these situations I have illustrated as scenarios and also identified how we troubleshoot in each case. Scenario number one is that the quality control did not perform as expected in both the C6 or the C6 flow cytometer. The solution in this case is it's extremely important that we know the correct beads that are being used for the flow cytometer. If we are using the C6 flow cytometer, we must use the 8 peak beads, and the gating should include 75 to 80% of the events, and the coefficient of variation of the brightest peak should be less than 5%. If we are using C6 plus flow cytometer, then we do not use the 8 peak beads, we use the CST RUO beads, and the further adjustments can be accomplished using the zoom tool. Scenario number two is that a patient sample has a volume of 0.5 ml and a sperm concentration of 2 million per ml. How do we proceed? As I mentioned that we need 2.5 million sperm to perform this test because many sperm are lost with the washing steps. And in this case, the volume is only 0.5 ml, whereas we require 1.25 ml volume to have the desired 2.5 million sperm. So in this case, the TUNEL test cannot be performed because of inadequate sperm concentration, and we will ask the patient to provide us another sample. Scenario number three is that the positive kit control shows a very low sperm DNA fragmentation of only 15% instead of the 30 to 40%, which is the desired percent of DNA damage. How do we overcome this situation? The solution here is that this is a test failure because the positive control did not give the desired percentage of DNA fragmentation. We make sure that we have used the correct template. We performed the staining steps correctly, the reagents were prepared correctly, and because it's a test failure, we should repeat this test again. Scenario number four is we have very few events which are recorded, or it is taking a long time to record 10,000 events. How do we proceed in this situation? The solution here is either the number of sperm in the sample are too low because they have been lost during the washing steps, or the sheath injection port or the SIP is clogged, and therefore the events are not being tracked by the laser. And in this situation, we will follow the back flush step and also clean and unclog the SIP and take the measurement again. So the take-home message for this test is that the standardized protocol, the quality control, the flow cytometer settings are extremely important. The test should be validated before it can be used as a diagnostic test. Based on our experience, we have created the templates, we have established the cutoff, and in our setting, a cutoff of 17% is what we use to determine the patient samples which have cutoff below this, or they are positive for sperm DNA fragmentation if they have a cutoff higher than 17%. And ideally, each lab should develop their own cutoff by testing a cohort of patients and proven fertile men. And this test, when used with flow cytometry as explained, is a very reproducible, robust test for measuring sperm DNA fragmentation using flow cytometry as a tool. So in the end, I would like to thank you for your attention. And this is one of our group picture and another view of the.
Lastly, you can connect with our center by sending a request, searching my name and Cleveland Clinic. And the benefit of connecting is that we are providing daily updates on our research activities. We provide free access to hundreds of research articles published by our center, by our scientists and researchers and our collaborators. You can also read the latest research which have been presented at important national and international meetings, reproductive meetings and urological meetings. Learn about virtual research opportunities that our center provides. Learn about our online ART training courses, and also the opportunities to collaborate with a team of ACRM researchers. So please consider joining on our LinkedIn.